Hello, I'm Ushma Neal with the Journal of Clinical Investigation, here to introduce a special episode of our series, Conversations with Giants in Medicine. Today we shift our focus and will be allowed to listen in on the conversation between three giants in immunology. Sir Mark Feldman will act as our interviewer as he talks with two of his best friends who happen to also be the winners of the 2019 Albert Lasker Award for Basic Medical Research. Doctors Max Cooper from Emory University and Jacques Miller from the Walter and Eliza Hall Institute for Medical Research in Australia were awarded this year for describing the function and existence of T and B cells, which have uncovered the organizing principle of the adaptive immune system.
Thank you, Ushma and the JCI, for this opportunity to take part in this video with two of my friends and heroes over the years. We're now long past that stage, and we're three of the elderly immunologists in our society, and we know each other well enough to have a full, frank and pleasant conversation. I first met Jacques the week I started my PhD in Weihai 50 years ago, in 1969, and Max a few years later, doing my first postdoc in London. He came for a sabbatical a few years later, so it's a privilege to be back together. So Jacques, you grew up initially, I understand, in China. Can you tell us a bit about your family and your parents?
Well, I grew up partly in China, partly in Switzerland, and back to China, and then to Australia. The reason why I went to Switzerland as a child was because my sister contracted tuberculosis, and Switzerland was the place where people said you could manage tuberculosis before streptomycin. But we had to leave Switzerland because of the German invasion of France, and we thought that the Germans would go through Switzerland. So we went back to China and stayed for 2 years before the Japanese war started. So we left China because of the Japanese. But my parents had no knowledge of medicine. My father was a manager of the Franco-Chinese Bank, and he was a very learned person. He spoke Japanese, he spoke all the Chinese dialects, at least 3 of them. He spoke German, he spoke English, and French, of course. And he was very pleased when I decided I should go into medicine. He just told me, go ahead, whatever you want to do. So it was a very nice familial atmosphere.
That's nice, yeah. Max, tell us about your family in the Deep South.
I grew up in rural Mississippi. I lived on a school campus. My father was the superintendent of this 12th grade school. My mother was a teacher as well. My father was a mathematician who loved learning, and he made sure that there were lots and lots of books in the school and in our home. So I grew up with books and reading them and learning about things around the world, and vowed that I was going to see some of that world when I grew up. They guided me. They had some problems. It was a little bit like being the preacher's son, to be the superintendent's son, and I had my full quota of mischievousness along with, but I did get instilled with a set of principles that were exactly the way my parents behaved, and a love for learning and reading that had made an immense difference in my life. One of my teachers in high school once told me, Max, if you hadn't had the parents you have, I think you would have ended up in jail. So family just that's where your real education and imprinting occurs to a greater extent than any other influence, I think, certainly for me.
So I thought that the real important topic to discuss and to lead up to is why immunology is now so important in the community and in medicine. And really the pivot of this discussion I'd like to start with is really how your contributions, your discoveries, your insights have led up progressively over years to the revolution in therapeutic immunology that's taken place in the last 10 or more years. So Max, do you want to start with that? What do you think has been the therapeutic revolution that you've seen?
Well, the immune system and its function impinges on everything that happens to us, everything in our physiology, whether it's your brains or your lungs or your kidneys, your skin. The immune cells go everywhere. They have so many interactions with other types of cells and with ones of their own kind that it's hard to think that there wouldn't be a lot of therapeutic implications if we understood well enough how these cells develop, function, and what can go wrong with them and how we can manipulate them. And thankfully, through the efforts of a lot of immunologists worldwide, we've learned a huge amount since the time that Jacques and I were stirring around defining these two major lineages of T and B cells.
I think there's a lot of awareness that the products of B cells, monoclonal antibodies, are very useful therapeutically, and that T cells can be engineered to kill tumors and perhaps other tissues usefully. What I think is not well appreciated is the fact that so many of our diseases of the Western world are in fact immunologically and inflammatory driven diseases. So this has been a very interesting conjunction that's made this revolution even more powerful. So Jacques, I remember when I stepped in the lab, you were a great conquering hero. You had published 4 papers in the JEM a few months before with Graham Mitchell. Can you take us through how you got to that stage, how your work led us to understand cell interactions in mice?
Well, yes, we were interested in the differences between mice that did not have a thymus from birth and normal mice. And we were able to quantitate the amount of circulating lymphocytes in neonatally thymected mice versus sham thymectomized mice. And there was a difference from 10 to the 8 in normal mice or sham operated mice to 10 to the 6 in neonatally thymectomized mice. So at the time I thought the difference was due to the fact that this thymus produces T cells that go out in the periphery and produce both cellular and humoral antibody production. So we tested that idea, and we found that the thymus-derived cells did not form antibody as we had thought, but the antibody-forming cells were derived from bone marrow, which we had used to protect adult thymectomized irradiated mice. So it was a very clear cut, and it suggested to us that there are 2 cellular major cells occurring in immunology, which are very important to produce various types of immune functions. But the T cells were helping the B cells to produce antibody, because B cells by themselves, in many cases, not in all cases, were unable to produce antibodies.
That's very interesting, but can you take us back even further, how you discovered that the thymus was an immunological organ when everybody else thought it was just a relic of vestigial?
Yes, that was done at a time when people did not think that the thymus had any function at all. They thought it was a useless organ, a graveyard for dying lymphocytes. And I was working on mouse leukemia which begins in the thymus and spreads. Mouse leukemia can be induced by a virus, but at that time the virus had to be given to newborn mice in order to get leukemia. And as a result of my work, I just decided I had to take out the thymus of newborn mice in order to investigate whether the virus could multiply outside the thymus. So take the thymus out of newborn mice, then give the virus, and then graft the thymus back sometimes later and see whether the virus has multiplied in the absence of thymus tissue. But as a result of this work, I found that neonatally thymectomized mice, that's mice thymectomized at birth, were very sick after a while, after about 3-4 months. And I wondered why, because it never seen sickness in adult thymectomized mice. So I looked at them, I did post-mortem, and I found out that they lacked lymphocytes, and they had lesions in the liver which suggested they had been infected by hepatitis virus, which is endemic in mouse colonies, but usually doesn't bother them. So obviously there was something wrong with their immune system, and I tested by grafting these mice before they were sick with foreign grafts from different strains of mice and even from rats. And to my amazement, they did not reject the grafts.
So these were very striking findings. Absolutely. And certainly underpin much of modern immunology. Max, you're credited with discovering the function of a bursa. Can you take us a bit through how these discoveries were made?
Well, a lot of people contributed to that discovery. I started out with an interest as a pediatrician in infections of patients, and in particular with children who were congenitally unable to handle infections, either having multiple bacterial infections, pneumonia, ear infections, or viral infections, and which were often fatal. And I was in Minneapolis with Bob Good and his group because we all had an interest in immunodeficiency diseases and how the immune system developed and what could go wrong with it in its function. There was a fortuitous discovery by Ohio State poultry scientist, by a graduate student, Bruce Glick, who found that on removing the bursa of Fabricius, a lymphoid organ at the end of the gut that was sometimes called the cloacal thymus, impaired development of antibody responses. He found it fortuitously because he started with the idea that it was affecting development, sexual maturation. It didn't affect either of those, but a postdoc came by, asked for some of his birds for an antibody demonstration, and made the discovery. That information and Jacques' information made us realize that it was particularly important to go back early if you were interested in what the thymus, and for the second organ, which other people had worked on, but the findings differed in different labs with different strains of chickens. So we did experiments really spurred by observations in our immune deficient children where the thymus and the small lymphocytes hadn't developed well, but they still had plasma cells, made lots of immunoglobulins and antibodies. And the idea was a single lineage of cells from Jacques' earliest results, that the thymus produced lymphocytes. Gowans had shown that they circulate around the body, and he, using a DNA label for lymphocytes, showed that they could become plasma cells, which were then known to make antibodies. So in order to test this idea of the thymus and the bursa being complementary or doing different things, we realized we needed to go earlier or reset the immunological clock. We did that by exposing newly hatched chicks to almost lethal irradiation together with removal of the thymus or the bursa of Fabricius, this hindgut lymphoid organ. And when they grew up and recovered from the irradiation effects, we found absolutely clear-cut results that indicated that the thymus gave rise to lymphocytes, were responsible for graft rejection, other cellular reactions. The bursa Fabricius was essential for development of germinal centers, plasma cells, and antibody production. And we could put back the bursal cells that weren't irradiated and restore in bursectomized and irradiated animals the whole B lineage, or bursa-dependent lineage. So that's sort of a short version of how back in the days, what many people call the dark ages of immunology, before the dawn of molecular biology, those were some of the clues that we put together and how complementary the work, our work was, Jacques and mine.
Absolutely, and of course with hindsight it's so clear that principles that are true in mammals and birds have really fundamental importance. But as I recall, the work that you did, Jacques and Max, was not initially that highly appreciated. So what led you to persist and carry on in the field but wasn't attracting so much approval?
When you say it wasn't appreciated, I think one should say the data were accepted.
Yes.
You could not possibly talk against the data when you see a mouse with 4 different skin grafts of different colors and even a red skin graft.
Yeah.
So you can't argue against the data, but you can argue against the interpretation.
Yes, that's what I meant.
My interpretation was that the immune system develops as a result of early thymus function. And their interpretation was that my mice, having been raised in converted horse stables, not in normal laboratory, because I was working out in the bush, so to speak. My mice, having been raised in converted horse stables, were so prone to infections that the additional trauma of thymectomy or irradiation and thymectomy in the adult would precipitate immunodeficiency. So that's what led me to go to NIH, courtesy of a Eleanor Roosevelt Fellowship, and repeat the entire work in germ-free tanks. And these mice cannot be infected because they're germ-free. We put skin graft on them after thymectomizing in early life, and the skin grafts were never rejected, even if they were highly disparate at the major H2 locus. So I think that convinced most immunologists that the thymus must have some function.
And I think it's a very good demonstration of the scientific method. If you get criticism, you address it with a new hypothesis and exclude it. So Max, what about the reaction to your work?
Well, it differed by different people, since it was partly based on disease observations in patients. Clinicians were a little more accepting than some basic immunologists, particularly because at that time there was not a wide appreciation of an evolutionary approach to understanding the immune system among immunologists at that time. I don't think Jacques and I ever doubted that the importance of what we had landed with the opportunity to see. It was just a question of how to answer some of the questions that our new view asked. If T cells didn't, or thymus-dependent lymphocytes didn't make antibodies, how did they see antigens? Where in the world was an equivalent site for generating B cells that were needed to make antibodies in mammals like us? And how did the cooperation actually occur? Or interaction, functional interactions between thymus-dependent and bursa-dependent, and as they got called, T and B cells quickly. So it was just so full of disease manifestations. It said you could divide leukemias, lymphomas into T and B cell malignancies. You could divide immunodeficiencies into one or the other. There were just so many implications that, I mean, we both were pretty excited about where to go from that point, although a lot of the questions that were raised took decades to answer, as you well know. And that was before the age of the cytokines and that you so wisely seized upon as a way of disease treatment with blocking their activities, these soluble factors that T and B cells use to interact with each other that we knew virtually nothing about in those days.
Yes. So continuing on this theme of talking to younger people, one of the things that's interesting is that you've done all your pioneering work in really basic science, immunology, but you both started in medicine. So Jacques, can you tell us a bit why you decided to study medicine first rather than go into science?
Well, I was influenced by the fact that one of my sisters contracted tuberculosis, and the fact that the other sister and I used to play with her, and we never got it. So I was wondering, well, what's happening? I mean, I overheard my mother talking to the doctor telling her, we don't know anything about tuberculosis, how it's done. So that sort of spurred my curiosity. I wanted to go to medicine partly for that, and partly for the fact that I was brought up during World War II when people were killing each other, and I just didn't want to kill anybody. I thought I'd better study medicine so I could patch them up rather than kill them. So, once I studied medicine, I became even more curious, because you see diseases like leukemia, you see diseases like systemic lupus erythematosus, and you want to know, why do these people get disease? Why don't I? Etc. So that spurred my curiosity, and I thought, well, the best way to do it is to do medical research.
Thank you, Jacques. Max?
Well, the local practitioner in my small rural town in Mississippi was one of the most treasured people in the community. Treasured for his knowledge, for his usefulness, and value in general. My father came from a large farm family, had ambitions to be a doctor, but didn't have the finances, he felt, to do that. And so he encouraged my early interest. As I learned about sports and girls and lots of other distracting pleasantries of life, and how long and hard I would have to study to become a doctor, my ambition waned a bit. My older brother was killed in a car accident. He was home, he was in the Marines, home on leave before going to Korea, and he was killed in a car accident. He had, we were very close. He left me as his beneficiary for his insurance policy. That took away my excuse of the financial aspects of becoming a physician. And it turned out that the more it was just such a lucky break for me to go into medicine. It got more and more interesting as I learned more about physiology, diseases, and it was just a wonderland for me. My particular interest in immunology came when I had decided on an academic career through encouragement of my pediatrics chief in New Orleans at Tulane. I by that time was very interested in immune deficiency diseases and allergy, and I went to take a fellowship. And on the way, I was asked to learn the immunofluorescence technique to study delayed type hypersensitivity. An immunologist in England took me into his lab to teach me the technique, and when he asked what I was going to do with it, I told him, and he said, well, as far as I know, this immunofluorescence technique is only useful for studying antibody production, not cellular immunity. I was so embarrassed that I decided I was going to learn, if nothing else, the difference between cellular and humoral immunity. I retreated to the library, found a book by your mentor and colleague, Macfarlane Burnett, on clonal selection theory, and it was written so well and in such an understandable fashion that I just was fell in love with immunology in a deeper sense, and so those were things that triggered me early on in my career.
And the love affair was reciprocated. Immunology's done well for you. So another theme I'd like to discuss is, as you make discoveries, what's the role of personal insight and what's the role of supervision? So Jacques, you were at Pollard's Wood, as you said, in the bush. So were you actually supervised when you were a student, or this was all innate, your own innate insights?
When I first arrived in Pollard's Wood, I was supposed to be supervised by Dr. Robert Harris. But within a few weeks, he left, not because of me, but because he was given a much better job at the National Institutes for Medical Research. So I was left without a supervisor, but I was able to inherit his animal space, which was very important because I was going to use a lot of mice. And then after having read what people did on mouse leukemia, I thought I could do exactly similar work in virus-induced leukemia, because virus-induced leukemia had just been discovered. So I thought, I have a great plan and I'm going to do it. But I did not realize that this would take me to immunology. So it seemed to work very well for me, and I didn't have to have a supervisor.
Max, were you closely supervised?
Well, when I joined Bob Good, I went in and said, well, here I am. What do I do now? And he said, well, when I went to Rockefeller Center, Macklin McCarty didn't tell me what to do. In fact, he left for a European holiday. He said, go in there and start working on some of those ideas you were telling me about when you said you wanted to come and work with our group. And so I exhausted some of those without making any great discoveries so quickly. But the freedom of feeding on the environment, everything that you can learn and what you can read, is a liberating way to start in anything. It's certainly not the only way to begin research. One has to start somewhere. So it's good to start on a project, maybe even one that's already well in progress when you start your investigations. There are lots of ways to skin a cat, and that's certainly true in immunology as well as other areas of biology, I think.
So Jacques, I remember the first year I was there visiting you in the animal room, seeing you do neonatal thymectomy with your technician and hardly being able to see the mice. So most people would not have thought that when they realized that a neonatal mouse is smaller than a fingernail, that this operation would have been possible. So what led you to believe that it was possible and actually succeed?
Well, the work on leukemia, I had to thymectomize adult mice. And I developed a technique which might be unique, I think, because I used to take out part of the sternum completely, knowing that the mice would repair that. They repair very easily. So I decided to adopt this particular technique to neonatal mice and do exactly the same in neonatal mice. And with my eyes at the time, not now, I need a microscope now, but with my eyes at the time I could see what I was doing, and a slight suction
Yeah.
very slight in a newborn mouse would do the job, and then you close the skin edges. You forget about the sternum because the newborn mice are going to renew the bones, and it worked perfectly well.
Yeah.
In fact, I thought neonatal thymectomy was easier than adult thymectomy because the adult thymectomy get pneumothorax very easily, but the newborn do not.
One important one is that from your vantage point, having had a wonderfully successful career, what advice might you give to young aspiring scientists on how to be successful and how to enjoy a career in science? Because that's one of the most important themes that I always see, that the enjoyment of science is fundamental to success and overcoming the hurdles. Yeah.
Well, I think you have to have some kind of hypothesis in order to investigate a certain part of your work. And if you have a hypothesis, you have to apply a certain amount of work. And I would say persistence, patience, and perseverance are the three fundamental points that we should have. In other words, a young person should, first of all, should be very honest in getting their data. They should be persistent to repeat the data and make sure it's correct. And they should be patient because it takes a long time to get a result these days. And they should be persistent in order to continue with the project that they have. They should, of course, be very curious to get a result and be absolutely in love with their work.
Okay. Thanks, Jacques. Max?
I agree with what Jacques says. Doing science and a scientific career is hard, but it can be so joyful. But as Jacques mentioned, you need to be in love with that type of career. And so starting out, if you wish to work in science, you need to find an area that interests you, that you're drawn to naturally. Find an environment that's successful. Find someone who has experience in science, or ones who have experience in science, whom you like and can interact with, and have a two-way kind of interaction.
Yeah.
And then you pick a particular issue that's not yet solved, or a particular technology that's needed even, and you address that, and that's the way you get a toehold, I think, to get started. And once you have any sort of success, it's like a drug. You are so euphoric to have learned something that you never knew before, that you're hooked for life. And I think that's what happened with Jacques and me.
Yeah. So Max and Jacques, you've had very successful medical scientific careers, but the world has strange ways. What do you think you might have done had this opportunity not presented itself? What other paths might you have found interesting?
Well, in my case, I would have done physics because I did very well at school in physics and mathematics. And I thought I could do astronomy. So I would have gone to astronomy. But then I thought, at that time, there were no satellites, nothing like that. So I couldn't imagine. I didn't want to end up teaching in a school, teaching physics. I thought, no, I've got something that has to excite me.
Yeah.
So I thought then the next best thing would be medicine.
Max?
Well, I started college, at a junior college, on a scholarship as a quarterback. Turned out I was the least capable quarterback among three others who were better than I was, and I was the slowest man on the team, including the coaches. So it was clear that I was never going to make fame and fortune as an athlete, although I was better at some other sports. And I loved to sing when I loved music, not in any great depth, but I just was thrilled with music.
Mm.
And my secret ambition, if I could have gone back and created my life, I would have been a saxophone player.
So I really want to finish by congratulating you on your lifetime of achievement, and that it's wonderful that the Lasker Committee has recognized your importance in science, and I hope you'll enjoy the next few days.
Thank you very much.
Thank you, Mark.
It's been a real privilege to be here today with you.
Thank you.
For us especially.